So in this video I will be talking about the Krebs cycle and how it occurs. So the Krebs cycle. So first let's get an overview. So the Krebs cycle occurs in the matrix of the mitochondria. And here it's an important thing to note that it runs twice per glucose molecule. And we'll get into why that occurs in a second. And finally, it requires oxygen to occur, but does not directly use oxygen, but it requires oxygen. And now let's talk about the whole process. Let me clear this. And so you start with acetyl coenzyme A combining with oxaloacetate acid. And remember from the preparatory step, you got 2 of those per every glucose molecule. So you have 2 acetyl coenzyme A's in total. But right now, to start with, you only need 1. So we'll get rid of that. And so what happens is acetyl coenzyme A combines with oxaloacetic acid to make citric acid. And so it kind of helps to know what the molecule is. And so acetyl coenzyme A is 2 carbons. Oxaloacetic acid is 4 carbons. And then so you combine them to make citric acid, which is 6 carbons. And so that gets converted into isocitric acid, and not many people talk about that step. That step isn't like that relevant, but this step right here, from isocitric acid to ketoglutaric acid, is important because the NAD comes in and oxidizes the substrate, and then so that releases carbon dioxide. And then ketoglutaric acid is produced. Now the next step, to make succinic acid, you do the same process, however with NAD+, so you oxidize the substrate, but now you're also producing ATP and putting in water. So ATP is produced by substrate level phosphorylation. I can't fit that down there. And so let's keep track of our carbons. So now this ketoglutaric acid is 5 carbons because you lost the CO2 there. And then you lost another CO2. Succinic acid is 4 carbons. And now succinic acid is oxidized again to make fumaric acid right here. Except it's oxidized by FAD instead of NAD. And then water is added to make malic acid. And finally, this is a final step, malic acid is oxidized by NAD plus to give your original product thing that you used in your cycle of oxaloacetic acid. And so let's essentially summarize what happened and what you put in and what you get. So per every glucose, so this cycle runs twice, and so I'm just gonna clear this and then write essentially a balance sheet, and so I'll keep that in the corner. So first you have 2 oxaloacetate plus 2 acetyl-CoA, and then 6 NAD pluses, and then 2 ADP and 2 FAD. Because remember, the cycle occurs twice, so therefore you use 3 NADs, and if it occurs twice, you'll need 6 of them for the cycle. And now let's talk about what we get out of it. So I'll write it down here. So what you get out of the Krebs cycle, or citric acid cycle, is you end up with 2-oxaloacetate, because remember, this just essentially just keeps going on. The oxaloacetate just keeps going in the cycle. And then you have 2 coenzyme A's, different from acetyl coenzyme A's. And then you have 6 NADH. Let me rewrite that. And then you have 2 ATP, and then 2 FADH2, and 4 carbon dioxide. And so now what you might ask is, well, what happens when you have enough of the carbon dioxide or ATP? Well, this whole cycle, the Krebs cycle, is regulated by competitive inhibition by oxaloacetate. So oxaloacetate competitively inhibits the enzyme required for the Krebs cycle.